This protocol uses laser capture microdissection coupled with RNA sequencing to obtain spatial and temporal transcriptomes from specific cells in plants of interest using small quantities of biological materials.
The main advantage of this technique is that it facilitates the direct visualization of cells within normal tissue context, allowing discrete cells to be precisely isolated in a contact-free manner.
Although this protocol is optimized for the isolation of plant cells, it can be applied to most cells that can be histologically identified.
Good sample preparation is critical. Therefore, when performing this technique for the first time, proper optimization of the tissue fixation and embedding is important before laser capture microdissection.
Before collecting the tissue sample, prepare a fixative appropriate to the species and tissue type to be harvested. For barley seed, cut the seed samples in half before submerging the samples into at least a 10x volume of ice-cold fixative. Use vacuum infiltration to accelerate the penetration of the fixative. The tissue should sink by the end of the infiltration. Then refresh the fixative for an overnight incubation and transfer the samples into cassettes for tissue processing. Place the tissue-loaded cassettes into the metal basket of an automatic processor and attach the metal basket to its holder above chamber 1. Set the program